Hello everyone, and welcome to our online lecture series for Bio2. Today we dive into our first truly meaty topic, that of meiosis. Because last time, hopefully we did a sufficient review of the processes of mitosis, the process of cell division that results in having identical products, meaning that the two cells created at the end of mitosis are exactly the same as the one cell in which you began. Now, we discussed all of the phases that are necessary for this to occur. In interphase, you have G1, where the cell grows, S phase, where the DNA is replicated, G2, where the cell also grows and accumulates more resources, and then you begin that process of mitosis with prophase, the condensing of the chromatin to chromosomes, prometaphase with the formation of the kinetochores and the movement of these chromosomes, metaphase where they're all lined up along the middle, anaphase where they split, and telophase, which undoes all the different components of prophase. Now, looking at this from the perspective of ploidy, we know that a diploid cell, a 2N cell, will replicate all of its chromosomes to result in a 4N cell temporarily, where each of those two sets of 2N are identical to one another. Because ultimately, mitosis will result in two cells that are each 2N and matching perfectly. Once you understand this basic process, you can see how meiosis will work. The bird's eye view on meiosis is that rather than making identical products, you are making four unique haploid cells, meaning that their ending genetic material is a single N. And it's critical that you understand that all of these will be unique at the very end. So we're going to go incrementally through this process so that way we understand first how to generate the haploids, and then by the end of this lecture, we'll be talking about how they end up completely unique when you've only got the two sets of DNA to begin with. Now, looking at meiosis from a view of ploidy, you have a 2N cell that will duplicate its genome to be 4N with those two sets of 2N. That two sets of 2N will then go through incremental steps, where first it ends up as two cells that are 2N, and then we'll eventually get to our final product of four cells that are each haploid and ultimately genetically unique, as indicated by the different colors here. So, what are these cells? Why do we want haploids? Why isn't everything just reproducing via mitosis? Well, it is ultimately for that purpose of reproduction. Meiosis generates cells that are called gametes. Gametic cells are your germ line. Ultimately, both of these refer to our reproductive cells. Like sperm and egg. Now, gametes are in contrast to the somatic or body cells that we have in us. So like your arms and legs, or this dog who's poking his nose at me right now, that nose is all somatic cells. They're all diploid. The reason why we want our gametes to be haploid is that ultimately they need to combine. And we do not want to increase the total amount of genetic material every single generation. One thing that you will hear me say over and over and over again in this class is how poorly most organisms on this planet tolerate variation in ploidy. It oftentimes is lethal. Something crazy to think about. If you don't have the right copy numbers of chromosomes, most of the time that ends up in either a miscarriage or abortion in the womb, in the development of the embryo, or early death, whether it be during infancy or even later in life. But this copy number variation poses a huge problem, not just to humans, but to most organisms on planet Earth. In order to really understand what's going on with our copy numbers and the genes underlying them, I want us to take a look at this full complement of the genome that we're going to be working with today. So here we have our two sets of chromosomes, one derived from mom and one derived from dad. For better or worse, I am sticking with the unfortunate conventional colors here. We oftentimes as a society associate pink with feminine and blue with masculine children. I don't like that particularly, but it'll probably make it easier to visualize at least. Pink is definitely for boys and blue is definitely for girls, y'all. Pink is quite a stunning, nice color on a shirt, I will say. But here we have the entire complement of our genome, where you have two chromosomes from mom of chromosome 1, meaning that you are diploid for chromosome 1, and two chromosomes, one from mom, one from dad, of chromosome 2, meaning that you are diploid for chromosome 2. So we know that these are derived from different parents. But how do the chromosomes know? Well, in truth of matter, they don't actually know what parent they are. They don't remember their ancestry. Just like if you weren't told it when you were young, you wouldn't know it either. But we can look at the features that are present in order to determine that ancestry. And in order to understand what I'm talking about here, we need to introduce probably my 3 favorite vocabulary words, at least of this lecture, if not of this entire class. The idea of a gene, the idea of a locus, and the idea of an allele. And I'm going to blow up an illustration of mom's chromosome 1 and dad's chromosome 1 in order to illustrate this process of genes, loci, and alleles. And yes, locus plural is loci. So a gene is a DNA sequence that makes a protein. Any protein. It's just coding. We'll talk a lot more about gene structure later in this class when we talk about how we actually make those proteins. But for now, all you need to understand is that a gene makes a protein and that it is a DNA sequence. The location of this gene on a chromosome is its locus. So, we can indicate the locus of a gene with a green box here. And we can say theoretically here that in both cases this is the gene for eye color on chromosomes 1 of mom and 1 of dad. We don't know what eye color is given, but we do know that eye color is present. The locus is oftentimes a base pair coordinate. So hopefully you remember from last lecture that these long chromosomes are actually millions of letters of DNA underlying them. And so we can say that the gene for eye color begins at the 150 millionth base pair of chromosome 1. Whether you're on chromosome 1 of mom or chromosome 1 of dad, the gene will have the same location. I'll say that again. The same gene will very often, can't say always because there's some exceptions, but the genes will often have the same location. What allows you to differentiate between mom and dad here is what the allele is. The allele looks at the version of the gene that you have. So what is the actual protein product? What is the end game? What does it make? For example, my mom has green eyes, and my dad has blue eyes. And so in this oversimplification that's not biologically real, but instructional nonetheless, I can say that my mom's chromosome has the green allele at the locus for the gene of eye color, and my dad has the blue allele at the locus where you find the gene for eye color. And in this way, in series of alleles, for example, you could potentially imagine that both your parents have blue eyes, and now I can't tell. But somewhere along this chromosome, there will be defining alleles that only your mom has or only your dad has. And that allows you to identify which chromosome derives from which parent. And that's ultimately because all of those different genes, and there are thousands of genes per chromosome, they're all physically connected to one another because they're found on the same chromosome. Those chromosomes that share the same genes at the same loci are our homologous chromosomes, meaning that they share the same genes and loci, but potentially not the same alleles, more than likely not the same alleles. In the process of meiosis, these homologous chromosomes can shuffle around the alleles. So that way, in some cases, you will get a mixture of mom and dad on the same chromosome. We'll discuss that by the end of this class. But that process of shuffling is called crossing over or genetic recombination. So with that vocab under our belt, let's do a quick review and a quick sketch of what things look like in the process of mitosis. And while we're doing this sketch, I want y'all to pay attention, very close attention, to the movement of the chromosomes. Because this is a very important topic as to the end products generated here. And I have a slightly embarrassing story during grad school where I was interviewing students to enter the grad school at UC California, Davis. And there was one particularly kind of arrogant student who had received a large fellowship and was thought very highly of himself. But, I didn't really let that sway my opinion one way or another about this individual. And I asked my general questions to see what their capabilities were, what their understanding was, how they could explain things, whether they would be a good candidate to work with, kind of an interview, because even as a student, you're potentially interviewing a future coworker here. And I asked this individual to draw out the processes of meiosis and mitosis on the board and explain to me what was going on with genetic recombination here, because that was one of the questions that I was working on in grad school. And he completely failed. Applying to a genetics program, he was unable to sketch out the path of chromosomes and the consequences, therefore, for recombination. I am determined to make sure that that happens to none of you at any point in your career. So therefore, we're sketching it out in great detail here and now. So, mitosis, just like meiosis, has an interphase, where in G1 the cell is growing, you have the full complement of chromosomes, they're unwound as chromatin. In S phase, each of these halves are duplicated. So that means that whatever the gene here is the same as the gene here, but also whatever the allele here is the same as the allele here. These are our homologous chromosomes. The alleles will be different when you look at the same locus on your chromosome from another parent. In S phase, everything is copied, DNA replication, and note that I will be indicating chromosome 1 is different from chromosome 2, in part because of shape here, but that's an artificial distinction to help us keep track of things. Know that the cell itself is not actually labeling any chromosomes anything. In G2, chromosomes pretty much stay the same. The rest of the cell is doing its thing. Once you hit mitosis, you hit prophase, the first step where our DNA is condensing. Oop, my blockout feature was a little bit too much there. The kinetochores form in prometaphase, and in metaphase, things fully line up. Now, the lining up that I did here, and even here, is untrue. What we should be looking at within the nucleus, so if that's our cell and this is our nucleus, all of these chromosomes you have to make them real small, are completely overlapping one another and kind of fudged in the mix. Oh, even smaller here. Completely condensed, completely uncondensed rather, as chromatin. During G1, S phase, and G2, they start forming into the shapes that you would recognize, and then ultimately move to their locations that you'll recognize in metaphase. In anaphase, things split up, and in telophase, we undo the steps in prophase. And now this splitting, I'll go draw an exaggeratedly large nucleus here, so pretend that that is your cell and this is your nucleus. The splitting occurs in such a way so that all of the chromosomes, all of the chromosomes on one side migrate to their respective side, and all of the chromosomes on the other will migrate to the other cell. And keep in mind, when they are pieced together or connected together in a large X-shaped chromosome, these are two different sister chromatids. And it's sister chromatids that are separating in mitosis. So at the end of things, you end up having a 2N cell next to a 2N cell, and everything is identical and happy to continue living on. Let's compare this to meiosis. Nope. We'll introduce the tetrad word in just a second. I don't want it up there just yet. So, in meiosis, interphase looks exactly the same. You've got all the two sets of chromosomes in G1 that will be copied in S phase that continue chilling in G2. And again, remember, just like before, they're actually sitting around as chromatin at this point. Homologous chromosomes made up of two sister chromatids will be what we're tracking when we get into the two different steps of meiosis. Now, meiosis has a prophase 1, prometaphase 1, metaphase 1, anaphase 1. Hence, you've got a P1, P1, M1, A1, T1. But because one round of this division ends up in a cell that's diploid, 2 cells that are diploid in this case, you need to do the whole thing over again in order to end up with 4 cells that are haploid. Oops. And there are different ways that these things can be organized to result in different end products. So, in our first look at things, you can imagine that each of these homologous chromosomes are lining up not vertically on top of one another to split them evenly so that way each cell gets the same genetic material. They're lining up adjacent to one another. And then different chromosomes on top. So all of chromosome 1 will be in a different row than all of chromosome 2. This structure is called a tetrad, because there are 1, 2, 3, 4 sister chromatids. Even though they're not all sister to one another, they have the same genes and loci, but they might not have the same alleles. They're all lined up next to one another. Now within the cell, they will split in such a way so that way everything on the left goes left and everything on the right goes right. Just like you would with the sister chromatids. But this time, rather than individual chromatids going to each side, the whole chromosome is. So that way, each of the diploid byproducts from meiosis phase 1 is going to be genetically unique from the other. You might get things lined up in such a way that all of mom goes to one side and all of dad goes to the other. And now, in prophase, prometaphase, and metaphase, anaphase, and telophase 2, you will again line up just like you would have in mitosis with sister chromatids going to opposite cells. And yes, directionality does matter, so the left stuff will end up on the left and the right stuff will end up on the right. But in this way, you're getting a series of all mom, all dad, all dad. And each of these cells are haploids. Instead of having the 2 sets of chromosomes that we started with, they each have 1. Now I'll take this moment here to emphasize that even though this looks simple, I want you to take the time to actually draw these out. If you have colors, colored pencils, colored markers, gel pens, whatever, draw it out using colors. It gets complicated very quickly. So let's make sure that we understand these processes by physically drawing them out on your notes. Now, this organization of mom on left, dad on right is not a foregone conclusion. In fact, it's actually completely random whether mom's on left, dad's on right for each chromosome. You could get a second meiosis of the same cell that results with a mom 1 on left and a dad 2 on left. This would result in cells that are a mixture of all of mom's 1 and dad's 2, or dad's 1 and mom's 2. And again, these cells, when going through the second phase of meiosis, the second round of PMET, would shuffle in such a way so that each cell is again haploid. This shuffling, where it could be randomly assigned mom on left or dad on left for each chromosome, is known as the law of independent assortment. These chromosomes assort independently of one another, meaning that mom 1 doesn't care whether mom 2 is on the left or mom 2 is on the right. It's just going to coin flip whichever direction it goes, because remember, these chromosomes don't know what chromosome they are. They don't know which parent they came from. They just know which is their homologous pair. And in this way, you can shuffle things around. So you already see how you can get new genetic mixtures, even though, note, that these two cells are completely identical. So you've got all of dad and all of dad, all of mom, all of mom. And even though you've got shuffling around down here, these two gametes are still identical. These two gametes are still identical. So we have to introduce another step in order to ultimately generate completely unique gametes. I would be remiss if I did not mention the second law here, even though it's not particularly well illustrated. But it is basic. It's the law of segregation. What the law of segregation says is that at the end of meiosis, you will ultimately get only a mom or a dad chromosome or allele for each of your two diploid states. So basically, for chromosome 1, the law of segregation, chromosome 1, the law of segregation says that each gamete is going to be either mom or dad, never both. Unless things have gone awry. But that's another lecture. So they are segregating. They're separating, right? Mom and dad alleles are separating. The alleles at chromosome 1 are separating from the alleles at chromosome 1. The alleles at chromosome 2 are separating from the alleles at chromosome 2. And these are your two laws of genetics, foundational. And it was that law of independent assortment that that student that I referred to earlier messed up. He did get a full ride to Stanford, by the way, so that's kind of crazy. Just goes to show you that even if you are intelligent, even if you are well-funded, you still have to pay attention to the details. He got the full ride to Stanford, but based on a large number of our feedbacks, did not get accepted to UC Davis. So, let's talk about the variation that has to happen, the crossing over, in order to ultimately get us to our 4 unique gametes at the end of meiosis. I guess, one sec. I should say that the steps that are happening in prophase, prometaphase, metaphase, anaphase, and telophase are all the same as in mitosis. The only difference is that shuffling at metaphase. That's why I didn't write anything underneath the PPAT, is because all those steps are essentially identical. And same goes for PPMAT round 2. Those steps are the same, with chromosomes condensing, nuclear envelopes disappearing, nucleoli disappearing, spindle fibers forming in prophase, kinetochores forming in prophase, prometaphase, metaphase things lining up on the metaphase plate, but again, lining up differently than we saw. Instead of 1 deep, 4 long, these chromosomes lined up 2 deep and 2 long. And we ultimately can generate novel genetic diversity, truly novel genetic diversity, in this process of crossing over or recombination. Now, recombination is where you have your homologous chromosomes, so chromosome 1 of dad, 1 of mom, and they will exchange small pieces of DNA. In this case, you can see that there was some sort of cut along this. Mom's piece, mom's alleles, mom's segment, popped over to dad, dad's segment popped over to mom. So now, if I were to take this chromosome and separate it, you can see that there are different components made of each parent. I'm highlighting the recombined region in black, so that way you can see it, but ultimately you know that the highlighting is not actually there. That's just to draw your attention to it. This is how it'll look in terms of chromosome painting, but ultimately, if we're being truly accurate, it's just DNA at the end of things. So, we can tell and we color to trace stuff, but know that it's all still DNA. Now, the way in which recombination occurs is that the DNA does need to get cut. You need to break the DNA. That DNA breaking occurs because of the formation of something called a chiasmata. You can also see them referred to as chiasma. Doesn't particularly matter which one, but two names for the same thing. Now, these chiasmata or chiasma are structures, protein structures, that form between these homologous chromosomes. They're absolutely essential for the process of meiosis. In cases of meiosis where chiasmata are not forming properly, we oftentimes get errors in the process. Things don't split correctly. Things don't divide up during anaphase like they should. So chiasmata are actually essential components of meiosis. Now, these chiasmata fundamentally are formed because of protein structures between the two homologous pairs. They will form something called a synapsis. That's when the two pairs are lining up very, very close, and the synapsis will lead to recombination, or that breaking apart, that crossing over. That synapsis occurs, that recombination occurs, because in a structure called the synaptomenal complex, you have a series of proteins. And proteins, unfortunately, have another name. They are the MRX complex within the synaptomenal complex. This MRX complex is actually what's going to cut, switch, and repair. Whoops. The broken sets of DNA. That switching over occurs because of this MRX protein complex. It's a 3-protein complex. There's 3 different things in there. The M, the R, and the X. Not any acronyms that y'all need to memorize by any stretch of imagination for this, but know that they are part of the DNA repair pathway. And so fundamentally, because these chiasmata are forming, in order to have synapsis occur when the chromosomes line up close enough to switch over, where the recombination will occur at the synaptomenal complex, where a very specific protein complex, called the MRX complex, will cut, switch, and repair over the 2 different sides. Anytime you are breaking DNA or cutting DNA and repairing DNA, that process is inherently mutagenic. And we refer to any of that cutting of DNA as a double strand break, or DSB. And DSBs are inherently mutagenic because some percentage of the time you won't repair back properly. Things will not connect back correctly on a DNA level. So that means like maybe an A switches to a G and there's a mutation in the process of recombination. So recombination generates genetic diversity in two ways: by swapping alleles and by mutations in double-strand breaks. Now, these synaptominal complexes and chiasmata are much more likely to form in euchromatic regions, particularly the euchromatic arms of chromosomes. And in humans, you will actually get one chiasmata forming per chromosome arm. And we refer to a chromosome arm as the region, as anything above or below the centromere. So one chromosome above, one chromosome arm below. So each of our X's, or major X's, have two different chromosome arms. And you are, on average, per meiosis in humans, getting one chiasmata and one recombination event per chromosome arm. So that was a fair bit of vocabulary, but with the vocabulary down, let's do the drawing out to see how this works out practically once we combine recombinations with our laws of independent assortment and segregation. So in PPMAT 1, you've got recombination having occurred. The homologous chromosomes are lining up too deep. They're segregating in anaphase, so that way, in this case, all of moms ended up on one side, all of dads ended up on the other, but the recombination enabled for different small segments of mom and dad to go with the other. Now, when they segregate into their four gametes, you will have one set that bears no recombination, one set that does, another set, grabbed that wrong, apologies for that. Hard to grab these tiny things. And now you've got four unique haploids. The sets of alleles for all of your genes in these chromosomes are actually different. And in this way, through one possible combination, you've got four unique gametes. Alternatively, you know that with the law of independent assortment, you could get moms on left, dads on right, or mom 1, dad 2 on left, dad 1, mom 2 on right. This would cause segregation in such a way that you get a mixture of moms and dads chromosomes across your different sets. And again, when segregating in prophase, prometaphase, metaphase, anaphase, telophase 2, these things would also generate four unique gametes. Now, as we mentioned before, you are getting, on average, one recombination event per chromosome arm per meiosis. So there's a lot of recombination happening in the human genome. The recombination itself will actually usually occur during the time frame of S and G2. So this is your recombination window. And it will have usually finished up before you get into the process of meiosis. And this is because recombination wants kind of unwound DNA. It doesn't want it to be as packed as densely as we see in the chromosomes when they are moving. Instead, it's going to rely on thinner strands that we get in the chromatin during S and G2. Now, your book also lays this entire process out with events and outcomes. So let's go ahead and read through this and make sure that we understand all of the vocabulary being used here. So it highlights the fact that during interphase, particularly the S phase, the chromosomes are duplicated, resulting in sister chromatids that are held together at the centromere. It does mention the fact that the centrosomes are duplicated, but we don't talk about that as it's not relating to the DNA here. The centrosomes are the conductors of your spindle fibers. We then see things pick up in prophase 1, where chromosomes are condensing, the nuclear envelope is breaking, homologous chromosomes bind firmly together, forming that tetrad, those 4 sister chromatids next to one another. The chiasmata form between non-sister chromatids, and crossing over occurs at the chiasmata. Now, I do want to draw attention to the fact that these chiasmata, your book says that they occur during prophase 1, slightly inaccurate. Most of the recombination events are occurring during S and G2, but if you say that they are occurring during prophase 1, and you're relying on your book for this information, I will not take away points. So your recombination window, in reality based on your textbook, is all the way from S, G2, to P prophase 1. At prometaphase, too late. Recombinations should be resolved. So those 3 time points are when you're looking at recombination occurring. In prometaphase 1, you've got homologous chromosomes attaching to the microtubules or the spindle fibers at the kinetochores. Then in metaphase 1, the homologous chromosomes will randomly line up on each side of the plate. Maybe mom's on one side, dad's on the other, or a mix mash, depending on how many chromosomes you have. Once they're all lined up at the metaphase plate, anaphase then splits up homologous chromosomes to separate. You will note that this drawing here also includes some recombination events. Note 1 per arm as the average, because you want those chiasmata to ensure the fact that that division occurs properly. Without chiasmata forming, it's more likely that you don't get proper splitting of homologous chromosomes separating. Your book draws attention to the fact that sister chromatids remain attached, but homologous chromosomes separate. Now, in telophase and cytokinesis, telophase 1, the sister chromatids arrive at the opposite poles, they start to decondense, nuclear envelope forms back, the cytoplasm is divided in a cleavage furrow, resulting in 2 kind of haploid cells. Even though those haploid cells have 2 sets of haploid DNA. For the most part. And that's because when we looked at things back in these cells, we saw that it was, for the most part, 2 copies of mom's information. A diploid requires 2 different alleles, meaning a mom and a dad. So you might be diploid for a small segment of it, but you're not diploid for the whole thing. So your book chooses here to call that 2 haploid cells, each duplicated. Hopefully that makes sense. Now, after telophase 1 has finished, you will go back through telophase, the entire process again. Prophase 2, sister chromatids are condensing, new spindle, nuclear envelope going away. In prometaphase 2, the kinetochores are attaching, then your sister chromatids are lining up. Now you're lining up in such a way so that way each of the sister chromatids will separate. In anaphase 2, your book is particularly specific on this, that the sister chromatids are pulled apart by the shortening of the kinetochore microtubules. So they basically just start to pull in that rope. And then, of course, at the end, we have 4 haploid daughter cells. They're generally referred to as daughter cells. And now that is the process of drawing out meiosis with recombination. Know that I will expect you on the exam to be able to draw this out. I might tell you to draw this out for 3 chromosomes for which the organism is diploid. I might ask you to draw this out for 4 chromosomes that are for which an organism is diploid. It just changes how much you're drawing, but not these fundamental processes of the law of independent assortment and the law of segregation, nor these processes of forming chiasmata, forming those synaptometal complexes in which you have the cut, switch, and repair proteins of the MRX complex that repair your double-strand breaks that are inherently mutagenic. Now, I've repeated things a lot this lecture, and in all likelihood, I won't be doing this as much in the future. I kept saying things over and over and going over things so many times because it is very difficult to understand our future topics if this is not clear to you. So I include this in week 1 because this is the foundation upon which we need to build. You need to understand how to take one step before you run a mile and run a marathon. So please do take the time without your notes to be able to draw the process of meiosis out, just as I have done with all these different chromosomes, and move things around. Make sure that you can get to a state where you have 4 genetically unique haploid daughter cells.